Yeah, so we broadly we're interested in understanding the translational control of gene expression. So why is it that some RNAs are translated better than others, and why is it that you might have one RNA that's translated much better in some conditions and not well at all in others? So we use a variety of techniques to measure and perturb translation. Um, and one particular challenge we run into is that many of the core proteins that are involved in translation initiation are essential. So we aren't able to, say, knock them out, or even if we deplete them, it's very quickly toxic to cells. So, one of the things we really wanted to do is a better sort of genetic approach to very rapidly deplete some of these core proteins so we can look at the immediate effects of their depletion, and that will give us a better understanding of what they are normally doing in the cell during translation. So that's what we've been working on this spring, is a way to very rapidly deplete these proteins, and this is sort of co-opting actually a system that plants use to rapidly degrade certain proteins in response to plant hormones. So we can transplant that whole system into budding yeast, which is one of our favorite model organisms to work with. And then in yeast we can rapidly target some of these proteins, like the cap-binding protein that binds the 7-methylguanosine cap that's present on all RNAs, and target that for rapid degradation, and that lets us see what happens to cells when that goes away quickly. No, I think it's a wonderful program. I guess, certainly from my perspective, it was great to have Lydia here visiting the lab. I know that she got to work very closely with an experienced postdoc. I know that he really enjoyed that opportunity, and I think that she really got a lot out of that, an opportunity to see how we do science, and yeah, I think it's really great. It's really been a pleasure to have Lydia here. I think that she has really made some real progress in a project that's been going on in the lab, something that is actually really a pretty central interest of ours, something that we've been working on a while, and she's really sort of moved that forward, and it's been great to have her around and to interact with students and postdocs in the lab too. So I think she's really brought a lot to the lab too. It's been great to have her.